Well, thank you, Emma. I'm David Goodman-Meza. This is my first time at INHSU. It's my first time ever being a rapporteur. It's actually the first time I've ever stayed till the end of a conference. So a really lot of firsts for me. And before I go through the science, I want to give you a little bit of the wrap-up of my journey. So, what do you do when Jason Grebley calls you at 5 p.m. on a Friday and asks you to summarize INHSU in 10 minutes? And so you take a deep breath and you say, fuck yeah, that's what I want to do. This is me. So, dream job. So this week at INHSU, I really had a great time. First time in Africa. Went to the botanical gardens, went to the wine tastings, did Table Mountain. Last night, went to the Cabo Beach, saw you guys dancing on the dance floor. Jason couldn't be stopped. Really good night. And this is me today. Just like, F me. Pulling the slides, pulling the pictures, trying to get it all together, right? Similar to people before me, I kind of broke it up in themes. I took a mosaic plot. I had the much easier task of only having to summarize 30 abstracts, so thank you, Jason. As you can see, really, hepatitis C really remains the center of the clinical research portfolio. Half of the studies really are focusing on treatment efficacy and clinical trials of hepatitis C medications and test and treat strategies. Although beyond hepatitis C, right, we're seeing a body of work around harm reduction, opiate use disorder, and other pieces around HIV, bacterial infections, getting people vaccinated, and stigma and social determinants of health. So really showing the diversification of the clinical research beyond viral hepatitis, and where I think we really need to invest more and see more here. This map really shows the geographic distribution of the presenters, not necessarily where the work was done. Really the strongest representation comes from Canada and really reflects the leadership in community-based harm reduction and integrated hepatitis C models that they have there. We also see significant contributions from the United Kingdom, Australia, United States, and really applaud, hands up to our host, South Africa. Thank you for clapping with me. Other submissions came from Malaysia, India, Nigeria, really bringing important perspectives from emerging networks such as Africa and Asia. Notably, no abstracts really from Eastern Europe or Latin America, and I think this is really a gap as INHSU expands next year we'll be in Prague, and so we've got to figure out how do we get people to present their clinical research findings, right? So let's get into the rapid review. I really apologize beforehand if I butcher your name or butcher your findings, but it is the last presentation and you won't see me again. So, Stork C was presented remotely by Catherine Chappell from Pittsburgh, and I think this is really a game changer, right? She's evaluating the use of Sofvel for hepatitis C treatment during pregnancy, and this is really an area that as clinicians, we know what we should be doing, but it's not in the guidelines, so we kind of don't do it, and this is going to really solve one of those gaps. She presented an interim analysis where she enrolled 70 participants, 62 completed therapy, 54 had data for their SVR12. Efficacy results outstanding, near 100%. The two people that failed really had barriers to adherence. More importantly, treatment was safe in these women. Median gestational age was 38 weeks. 12% had preterm birth, but this is no different from women who have hepatitis C that are not getting treatment. No cases of cholestasis, no cases of vertical transmission. So really incredible findings. Takeaway, Sofvel was safe, effective, and could prevent cholestasis of pregnancy and perinatal hep C transmission. Chen Huat Kit from Malaysia brought us results from a sub-analysis from the EAST trial. They compared 12 weeks of Ravidisvir versus an 8-week course. And over 300 participants, really, they showed that the 8-week course was as effective as the 12-week course. But really specific to their sub-analysis that they presented was that patient-level characteristics did not matter in who achieves SVR or not. So if you report using drugs or not, you're as likely to achieve excellent results. Brian Conway from Canada, he presented 2 sub-analyses of his work with GP in acute infections. He really squeezed the juice out of that same data set. One analysis looked at people at their first infection versus previous infections, and other sub-analyzed people who had recent reports of injecting drugs or current injection drug use versus those who did not report. You can see again, excellent results, top-notch efficacy, liver function tests improve, bottom line, treat Hep C. Right? Not news to the choir. Okay, test and treat. So Jason's study, he presented findings from the TEMPO study presented this morning. This was a randomized, a cluster randomized trial that compared 3 testing approaches. Standard of care, where you get phlebotomy and then you send it off and then it comes back a week later, right? Dried blood spot testing and then point of care RNA testing. And really, if you look at that, the colored bars at the bottom really tells the whole story. At 12 weeks, point of care RNA testing, nearly triple treatment initiation within 12 weeks. And so we can really see in the little line charts above, the cumulative incidence curves, that this strategy really got people starting treatment much faster. One surprise, and this is what Jason emailed me, you gotta say this, was that the initiation of hepatitis C treatment was really much lower than what we would expect, right? We really want higher numbers. And so there's really a lot more work that we need to do to figure out how we can maximize this great strategy. Okay. In the next 2 sections, I'm just gonna try to highlight some other work in the research lifestyle and get away a little bit from the RCTs. So Kristina Nidzilova from, I butchered it, I'm terribly sorry, from the University of Dundee presented this pilot study where she explored whether wearable respiratory monitoring device that they call the PneumoWave Resp meter can help prevent drug-related overdose deaths by detecting early signs of overdose, which is a decrease or respiratory depression. The device is really incredible. It uses a small electrode that you stick on to your chest, and it detects people's breathing patterns. In this early evaluation, they were able to show that the device can detect patterns of breathing or apneic events, right? And then through qualitative work, they showed that people with lived experience of overdose viewed the device as acceptable and motivating, and of course, potentially for its potential to alert peers or services. Daniela Sephora from Johannesburg, she explored preferences from low dead space syringe and needles, really laying the groundwork for a successful rollout of this intervention. They did 3 focus groups among 25 participants, where they examine what matters most to people when it comes to syringe design, access, and safety. In brief, they described the barriers to safe injecting, inconsistent syringe supply, limited outreach days, frequent police harassment. They strongly preferred 1 needle fixed syringes. They feel it's thinner, less painful, and familiar. They feel detached options are intimidating, too clinical, and sometimes associated with veterinary use. I'm really loving to see how this project evolves over the future ensues. Within opiate use disorder, a local project here in South Africa in the opiate use disorder treatment space, Elaine Seaman from Sadiba Hope Medical Center here in South Africa evaluated outcomes on an opiate agonist treatment therapy scale-up and how it contributed to broader health outcomes. At the beginning of the program, only 1% of people who inject drugs access OAT. At the end of the program, only 4%, not groundbreaking, but it's a start. But I really think the striking piece is down below where we can see really the rollover effects to other health conditions, be that HIV, other viral hepatitis, and tuberculosis. So really, I need to close. I've got a big red thing. The light doesn't let me see exactly how many seconds I got, but I know it's red, so I'm kind of getting to the end. But it's the last presentation, so I think I can just keep talking and rambling. So the good really is obvious, right? The clinical portfolio at INSO really reflects a field that is moving from demonstration to delivery right. We have strong community partnerships and top-notch science really guiding this real-world impact. The challenges, I wish I took out the bad, but really this lack of representation, right, from certain areas. And as I mentioned, as we move to the next Institute of Prague, we really have to figure out strategies to increase the clinical research from these areas. Related to content, I really think we need to expand what's after hepatitis C treatment, how do we prevent reinfection, how do we improve the health of people using drugs. We really need to invest in long-acting agents, both for medications for opiate use disorder, for PrEP, antibiotics like dalbavancin and ritonavir for bacterial infections. I really saw little clinical research around polysubstance use, stimulant use, and other routes of using drugs like smoking or snorting. And then really a focus around First Nations populations, prisons, and women, where we really need to be having targeted interventions. So finally, thank you, Jason, for the opportunity. This is really incredible. I really enjoyed staying to the end of a conference, and I'll see you next year.